Hello. Today something related to the research on the origin of the coronavirus affecting 2020 worldwide, also called COVID-19. And the first of the articles that I want to mention is one sent as a letter to the editor that is called the Proximal Origin of, and this is another technical scientific name of the COVID-19, SARS-CoV-2. They just try to put a more technical name, and this is all the people involved, as you can see in the screen. Let me check if everything is working. It seems that the volume is very low, so I seem to need to speak louder. But I was mentioning people that sent this letter are researchers from La Jolla, California, the UK, New York, Australia, New Orleans, and even a biotechnology company by one of the authors from Maryland. I think that is a conflict of interests to belong to a biotech company and at the same time receive grants from the government and at the same time to publish an article. But let's see what is the most important aspect of this article, and I think is the image. I hope you could see here the image, and I'm going to try to explain as brief and as simple as possible. They say that the COVID-19, that they call it here human SARS-CoV-2, has a sequence that has not been found in any other living organism, and is composed by 4 amino acids, PRRAR, which means proline, arginine, arginine, alanine. And this particular sequence that they explain that is within the spike region, a region that has been the most analyzed from the beginning. They explain that it is because not so many animals have been sequenced. We still don't know the origin of that region, but by having a proline there in the protein, that makes a twist, a turn, a bend, an angle, and that allows for the attachment of other substances that make it most highly infective. So that's the unknown region, the black hole, the dark mystery right here. And the other situation is that when compared with one of the domains of this spike region, because the one that we saw first was called the polybasic cleavage site. Now we are going to see the receptor binding domain that makes it very, very, very infective in the sense of the spread, not of the damage, in the sense of the spread. even if it's causing a lot of damage, is not as dangerous, they say, as others that are more really mortal or more toxic, we may say. But the receptor binding domain, when we analyzed it, it has basically 6 specific amino acids that are vital for the receptor. There is here something that looks like an L, something that looks like an F, a Q, an S, an N, and a Y. That stands, the first for lysine. You can do all that study in detail by yourselves. The F, the phenylalanine, then the Q stands for glutamine, another amino acid, and then the S stands for serine, the one that is the serine, and then the N, it stands for asparagine, very similar to the glutamine, asparagine, and the Y, a big amino acid, stands for tyrosine. So these 6 amino acids are basic for the attachment to humans, and that's what makes this virus very extremely highly contagious, because of these 6 sites. So when they analyze the sites from bats, they discover that the bats don't have the 6 sites. So that's to discredit that the bats were the main responsible, even if a high percent of the gene of this COVID-19 seems to be coming from a bat. They say here that approximately 96% of the gene comes from a bat, with the key regions that make it highly infective, contagious, are not present at all in bats. But exactly the 6 needed amino acids for the attachment to humans, for the high infectivity, are present in the pangolin. Exactly the 6 in the exact positions of the COVID-19 that is attacking humans. So let me show you the pangolin. Here the pangolin, it's an animal that looks almost like a very strange and cute and small animal. That's the pangolin. And you can investigate by yourself the kind of bat that is being compared also, which is the Manis javanica. It's an horrible bat with a very flat nose and with ferocious teeth. But even if 96% of the COVID-19 seems to come from a bat, the specific key regions for the infectivity and contagiousness are not from a bat. The attachment to humans that make him highly contagious is not present in bats, but in pangolin. And the area that makes even more greater the infection within a human, once it's attached to his lung cells, is a region of 4 amino acids that has not been found in any organism thus far. Now, what is very interesting to me is that since 2015, researchers were presenting and doing research in their labs to analyze how the coronavirus could be more and more and more and more infective to humans. So wait a minute. Scientists were trying to make highly infective the COVID-19 since at least 2015? Yeah. For example, this article from 2015, published in a very prestigious journal, Nature Medicine, is talking precisely the potential for human emergence. So they were doing everything in this laboratory to make it more and more infective and to see how it will be more and more toxic. They were even talking of those two regions that we saw that are specific for the contagiousness and the infectivity in humans. And that's precisely what in 2015 this article explains. I quote from this article of 2015, spike adaptation in a secondary host is seen as a necessity, with most mutations expected to occur within the RBD, thereby facilitating improved infection. They knew how to increase the infectivity of this virus, at least since 2015. And if they were mentioning that, is because they were working tirelessly in most of the virus labs that were working with these coronaviruses, trying to see how it could be more and more and more infective. So that is very interesting. And the RBD means receptor binding domain. Receptor binding domain. So all of this was well known, how to increase the infectivity of this coronavirus. And they were even mentioning the kind of animals that were the best mediators, something that the media has not even mentioned. Let me read you another fragment of this. Building on this finding, the common emergence paradigm argues that epidemic SARS-CoV, exactly as they call the current one, just -2, originated as a bad virus, jumped to civets, so they knew that the civets were the mediator, and incorporated changes within the receptor binding domain to improve binding to civet ACE2. So, which is exactly what we are looking in those two regions. So the key words for you to research by yourselves is RBD and ACE2, a well-known fact since at least 2015. So when we search who were the researchers very involved in realizing how this virus could be more and more and more infective, and we need to remember that Bill Gates was warning people about this kind of research, and Bill Gates was saying these labs need to have a security measure as they do the emergency trainings for terrorist attacks. They needed to put safety measures of emergency in case one of these viruses escapes from the lab. And what is very interesting is one of the members of this 2015 article is precisely somebody from China, Wuhan, where the virus originated. Key Laboratory of Special Pathogens and Biosafety, Wuhan Institute of Virology, Chinese Academy of Sciences, Wuhan, China. And the individual that was involved in this research to increase pathogenicity of the coronavirus was called Xing-Ji. So if I were a reporter, I will be asking him and all the members involved in this paper what they knew and how they realized that this virus was being worked in a laboratory. See, most of the researchers are from North Carolina, Chapel Hill laboratories of epidemiology, of microbiology and immunology. But also we have researchers from Boston, Massachusetts, and the Harvard Medical School, both from the Dana-Farber Institute and the Department of Medicine, both of them from Boston, from the Harvard Medical School, and people from Switzerland. So if researchers knew and were working very hard to increase pathogenicity of coronaviruses, what does it make you think? Especially when you read this recent article of 2020, of February of 2020, and you realize that they were very clear that SARS-CoV-2, COVID-19, which is the same, seems to have a receptor binding domain that binds with high affinity to ACE2 from humans, but also binds with high affinity, or binds with high affinity to ferrets. Let me show you a ferret. So the ferret, not only the pangolins and the bats, is another high responsible candidate as a mediator. And researchers, of course, could be inoculating a lot of ferrets. And in a country as China, they could even be inoculating humans and paying them just to increase the pathogenicity of this virus. So these are the ferrets mentioned in that article. And what surprises me the most is that people is not talking about these other animals. Even cats, the daily cat and other species have a high affinity to the coronavirus, which means that are the biggest mediator close to home, and nobody's talking about cats being the mediators of the disease. And right here we can read that, and other species with high receptor homology. So I don't need to put an image of a cat, but you need to think about, if a cat is a mediator of coronavirus, why the researchers have not even mentioned until now me in the main stream that cats are very risky to have them around when there is a COVID-19 infection, because they are the hosts of that virus to be spreading more and more around. And that is exactly what we read in the article of 2015 about a wild cat that is the civet, is the Siberian cat. Let me show you. Okay, I can close this of the ferrets. I can close this of the pangolins. But, well, I won't close the pangolin until showing you more of the text. But you saw on the image that the only one that has the 6 amino acids needed for the contagiousness in humans is the pangolin, not the bat. And let me show you the civet, Siberian cat. You see the Siberian cat, and the cats are the hosts chosen even before pangolins to be really, really highly infective and able to, with those 6 amino acids, to attach the coronavirus and get the disease. So why people is not asking the researchers that were seriously involved in increasing the toxicity of this virus? The same persons of this article, this menagerie, Vinit di Menagerie, this researcher, even published in 2019 that he have found a way to overcome the barrier of species for the toxicity through the use of tyrosine. He's from the Department of Epidemiology, University of North Carolina, Chapel Hill. So this is very intriguing, and because I don't want to go on and on and on,
I didn't quite catch that. Could you please repeat it?
I don't know why, while I was talking about this other phone got activated by itself. I'm sorry, I needed to turn off a cell phone that activated by itself while I was speaking. But this is very interesting to me. If the cats are one of the most common animals that receive the virus with very high affinity, why media is not telling stay away from cats, and why media is not telling that the civet was one of the most common experimental cats, a wild cat, for researchers to figure out how to increase the pathogenicity of the coronaviruses, specifically in those two areas, the ACE2 and the RBD. So you need to read by yourself the articles and question and ask the hard questions. Because, for example, this article, The Proximal Origin of SARS-CoV-2, has a lot of red points that I have no time to discuss with you, but I'm going to be moving it slowly for you to take note of the things that I put in red, for you to do your own research. But I just want to stop to read about the pangolins and then to conclude with this, because there are a lot of other things that the mainstream media is not really mentioning, as if they were not interested or manipulated by whomever controls most of the mass media in this world, because this is very obvious and interesting, and it's a bummer that is not being mentioned in the mainstream media. Some pangolin coronaviruses exhibit strong similarity to SARS-CoV-2, to COVID-19 is the same. In the RBD, including all 6 key RBD residues, is what we saw in the image right here. The thing that we saw right here in that what I call the hollow, empty, dark abyss. Then they keep talking about very interesting things that I have no time and really not so much into investing time in reading to you in detail all of these things. That's a shame. That's really sad. You need to read it by yourself. But those in red are points that really raise eyebrows about what are the methods to stay away of cats as the hosts for this virus that are facilitating the spread in humans, as they did in the lab with the civet and with many other organisms. Then there is a researcher that is a retired researcher that is into computers. In this case, he was an IBM emeritus, International Business Machines emeritus, and from the IBM European Research Center on Artificial Intelligence. Now he's retired. He was also a PhD in mathematics and in computer science for the Bordeaux University. So he's not just another idiot more, especially in his acknowledgements. This is another article that I just felt compelled to add. He says, I especially thank Professor Luc Montagnier, the discoverer or co-discoverer with Robert Gallo of the HIV. I especially thank Professor Luc Montagnier for his advice and suggestions that led to this article. So he is thanking Luc Montagnier, Medicine Nobel Prize winner, for their interest in my research for biomathematical laws of genomes. So he is not just another idiot. Even if he's retired, he keeps on publishing. Even if he keeps on publishing in small journals, he's doing his own independent research, and for that reason he really don't need and don't have any financial dependency of any institution, and he really is not biased by his belongings on any laboratory or biased with hiding information. So I'm just going to read a little bit of what he says. He says, it is very likely that there was human intervention in the region of this virus. Analysis of this region shows that a very small region has at least 9 fragments, well, 6 fragments, and in total in the virus 9 fragments of HIV fragments, another retrovirus, the HIV, and he shows here the homologies that are not present in any other virus as high as in the COVID-19. That is a fact that has 9 fragments of HIV genes within his genome, and 6 of them run in a very, very short region of almost 900 kilobases. And we need to remember that fragments as short as 12 nucleotides are important, like as linkers. So in this case, these fragments are a little bit longer than 12 nucleotides. This is one of an HIV. You need to check from the M protein. Another of the HIV of the M protein. That's the second. The third, another region of the HIV from the M protein as well. Another fragment in the COVID-19 of HIV genes, of HIV nucleotides. This is from the RT fragment, and here another region that I guess we're gonna search later, and even from the simian virus, SIV. This is from the pole region of that SIV virus, and those are the identities. So this independent researcher that is into the computers needs to mention here why the coronavirus gets so easily integrated into the human genome to be so toxic, so to multiply itself so much. He's saying here evidence of a kind of intelligent will in the design of this COVID-19. He says both figures proves evidence that the 6 HIV-SIV human immunodeficiency virus and simian immunodeficiency viruses inserts are a result of natural evolution and mutations, because they are in a very close tied region that he says that is within less than 900 bases of pairs of bases with 275. Firstly, the 6 inserts are highly contiguous within 275 base pairs regions. So now I understand why he put this fragment of the sequence of the coronavirus. He says in this fragment of 300 nucleotides, it is where the 6 human and simian immunodeficiency virus fragments were found by him. Nobody's talking about this, not any other researcher, even when we know that people were doing research how to make more and more infective the coronavirus, at least since 2015, including people from Wuhan and other labs. He says, firstly, the 6 inserts of the HIV are highly contiguous, less than 275 base pairs, or at least 275, less than 300. Secondly, HIV, SIV inserted strains are very homogeneous from Russia, from Côte d'Ivoire, from Netherlands and Malawi origins. Third, the functions of inserts are also various, 2 for pol-RT, 4 from envelope, ENV, and pol and ENV are 2 of the 3 most important for the infectivity of the HIV functional genes. Not reported in this article were also found within the COVID-19 genome 3 more HIV SIV regions, one from ENV, one from RT, reverse transcriptase, because it's a retrovirus, a reverse transcriptase, and one from integrase. So 6 plus 3, there are 9. And he says that's not random. That's not randomness, but that's a purpose. That shows the human hand in these 9 inserts from the HIV. These 9 inserts from the HIV in COVID-19. Pol RT integrase and ENV envelope. Pol int and ENV. We have here 3 major functional pieces of genes necessary to build a retrovirus. In other hand, symmetry strategy of realism, numbers of inserts by increasing frequencies and pathogenicity. Symmetry economy strategy, sense and antisense in an insert of minimum length. All this is remarkable and bears the mark of a desire for organization of a human nature. And here is his drawing that is showing the fragments in that segment, that very short segment of less than 300 base pairs in the COVID-19. I am putting the rectangle within the COVID-19 sequence, and let me expand it a little bit more. So this that I am expanding here is a little less than 300 base pairs, and he found here 6 fragments of HIV, which means 5 fragments of HIV and 1 fragment of SIV, simian immunodeficiency virus, 1 fragment, the last one in the right side, and the rest fragments in the left side of it, human immunodeficiency viruses within the COVID. As I say, the media is not talking about that. They are ignoring this. That clearly shows the human hand. He's also presenting it with the names of the sequences, the full names of the sequences, and the sequences themselves within those 275 base pairs right here. So you need to check this situation. His article is called Wuhan COVID-19 Synthetic Origins and Evolution. Evolution meaning that the adaptation to humans to be more and more infective. He published it in an Indian journal, International Journal of Research, and let me just read this to conclude. Ethical considerations. The 2 main results of this publication confront us with a paradox. In fact, number one, on the one hand, we have just demonstrated that this COVID-19 genome contains an insertion of 6 strategic regions of HIV-SIV concentrated in a mini space representing less than 1% of the length of the genome. One can think that such a disturbance can only have affected the global order of the DNA of this genome. Affected the order of the DNA of this genome. The researcher, the retired IBM emeritus and university PhD, is Jean-Claude Perez. Very interesting. You can find his full article in ResearchGate. This is very interesting, but you can also read by putting pause in what I am showing you here, because I'm gonna upload this slowly, as I will do with the other articles before I depart. So he says the 2 aspects. The second aspect, we have also just showed that since the first SARS of 203, also from China, the global order of successive genomes was only increasing, highlighted here by stationary digital waves calibrated on increasing Fibonacci numbers. This goes like woof above my head. This is a deep programming skill that to demonstrate that this COVID-19 have all the hallmarks of the human hand and not of the natural adaptation. In particular, COVID-19 is structured over a long distance via 21 base pairs of amplitude wave. That also goes above my head. However, the deletion in this genome of the small region, including the 6 HIV-SIV inserts, will further gain in organization, causing waves of 34 base pairs to emerge, the number of Fibonacci following 21 base pairs. So he's saying that with his profound programming techniques, he found out that if that region of the HIV-SIV were removed, the sequence in itself will be more symmetrical. That is, this is disturbing the symmetry and make it even the virus not as graceful as if that sequence were not there. So he's saying this sequence is there because a human hand inserted it for a specific purpose that goes beyond of the natural symmetries of organisms and things. This is causing a disturbance that is anti-natural. These 275 base pairs with sequences from the HIV. We must conclude this remarkable fact. To adapt to the disorder of its DNA, resulting from the insertion of the 6 HIV-SIV, plus 3 more in different regions, the COVID-19 genome has most certainly increased its level of global organization by adaptation mutations. And we will now have to fear that this genome will continue to mutate in order to optimize its overall level of organization, jumping back and forth between cats and humans, and changing a little bit even between human and human, to adapt and to be getting more infective is what he's saying. So people of the disinformation, like Sanjay Gupta knows that the kind of protectors that he's promoting, the flat protectors, rectangular, are worthless because the virus can enter through the sides. Even if his daughter is making those are worthless and he knows it, and he keeps promoting them. And the other thing that Sanjay Gupta is saying that this virus is static and not as highly mutating as others. But this kind of computer research shows that the virus keeps on changing, and also the previous research shows that. So I'm gonna just be moving it little by little for you to put pause and be reading this by yourselves. But I want to say that this article is very worth to be taken seriously, because these 275 base pairs filled with sequences of the HIV is not something random, not something natural, but anti-natural. These are the other sequences that he compared. And as I say, many things go above my head. All this analysis that he says that he compares some of the sequences. He even says, again, what we know, that Neanderthals were not our ancestors, but he, by doing a comparison of some situations their chromosome 4, but that's aside the point. So let me keep on moving this slowly for you to put pause and to keep on reading to get the whole article through this YouTube situation. So you put pause. I hope you could read it or contact directly the researcher, as well as the other researchers, the one from Wuhan that was working with an international team that were figuring out how the coronavirus could increase its infectivity into humans. And they were publishing that since 2015 without paying any attention to Bill Gates. So the programmers can be a good help to figure out the solution of this pandemic of the world, like this programmer that found this thing. Because once we know the origin, we can go and ask for the antidote to the originators of this kind of virus. Because he says that it shows a partial human hand that is abnormal, that region of 275 base pairs with sequences from the HIV. Now, the ridiculous claims of others are trying to discard 100% the possibility of human intervention. But they say that if it were natural, it would take a very big number of hosts and a very long time to go undetected in a very big number of hosts, and take a long time to go undetected in China. Kind of begs the question, like, are they thus so incompetent really, or something else is going on? So you just need to check for yourself, especially if you are a scientist with a scientific mind, and figure out exactly how to interact with the programmers and the ones that were more involved in developing strong strains of this COVID to find the antidote. You need to ask everyone and each one of the ones of the article of 2015 how to counter the effects of the COVID-19. They may be aware of that because they were working in ways to figure out the infectivity to be increased all the way since 2015, knowing about this RBD site and about the ACE2 sites. So you need to ask the questions and not only be all scared. And if possible, I may suggest stay away from cats during this time of the pandemic. Another way that shows that there is some sinister interest in the mainstream media that is not telling you that the cats, the ordinary cats that roam around your house, are the ones that are the intermediary hosts for the increase of the infectivity of this virus. He mentions a lot of other distinguished persons from all over the world that helped to this programmer to publish what nobody else that is in an institution dares to publish because of the status quo and other kind of stupid things that delay the real progress of the antidote. In my case, for example, I have been also working independently, finding out that the I Ching is really a computer program, the core of a computer program that can help also to counter the effects, as with this programmer. I'm going to close this one, by searching also the comparison of sequences with the strategies of the position of the 64 codons within the I Ching. The I Ching was one of the earliest representations of the genetic code before of the flood of Noah, according to my research, found in China by Chinese, who is a powerful research tool for genetics. These are all the guys that you need to interview to find the antidote against the COVID-19, because they were the ones that were working in how to increase the toxicity and pathogenicity and infectivity and contagiousness of the virus. So you can find by yourself. This is available online for everybody. The same as this other article is available online for everybody. So I pray God help as soon as possible to programmers applying their knowledge to the biosciences and to the ones that were involved in the study of these coronaviruses to give us the antidote in the name of Jesus Christ. Thank you, and I hope this at least awakes your interest to do your own research, because there are a lot of questions like this big abyss, PRRA, which source, if it was natural, was that, or if it was not natural, seems to be something artificial in another very close region of 3, 6, 9, 12 nucleotides in a sequence, exactly as the 12 nucleotides that when I did my research I found that were the linker that was from Echo R1 in a totally different research, contaminating thousands of sequences on the Genbank. This is a sequence as long as that, 12 base pairs, that are increasing the infectivity of this organism. So, well, there is a lot to say, but I let you do your own prayers and your own discoveries to find the solution, at least locally, for you in your house. Get rid of cats for now. Bye bye.